Hi everyone! We've reached our final lectures for the semester. So today's study tip is to tell your entire household the dates for your exams. Put it on your family calendar, your significant other's calendar, your housemate's calendar, because then they'll know that you're going to be extra busy studying the week prior to the exam and maybe even provide you extra support. So chapter 20 is about reproductive systems, and these are commonly divided into male and female reproductive systems, but I've changed that a little bit today to really prevent our focus on this sex binary, right? And we talked about in a previous lecture how sex and gender are not the same thing. But even for things like chromosomal sex, male and female, there's not just that simple sex binary, right? And so I've called these lectures reproductive physiology, and we'll talk about spermatogenesis and ovulation and some of these topics. But knowing that people that identify as male or female or non-binary are going to experience these things too. So as you think about providing care for a variety of people, I want you to think about expanding that idea of a gender or sex binary. Okay? So we're almost there. Time to make the final big push, put in extra study time, and finish strong. I know you can all do it. Today we're going to talk about chromosomes and meiosis, and these should be a little bit of a review, but I'll remind them because they're really important for this chapter. Then we'll talk about spermatogenesis, fertilization, sex determination, and hormonal regulation. Okay, so to remind you that chromosomes in humans, in our somatic cells, our body cells, we have two sets of chromosomes. They're called diploids, so 2N, and this is because each chromosome has a duplicate copy, okay? So if we look at, like, for example, chromosome number 1, you have 2 copies of chromosome number 1, you have 2 copies of chromosome number 2. You inherited 1 from the sperm that made you and 1 from the egg that made you. This makes you a diploid. In reproductive cells, gametes, you have 1 set of chromosomes, haploid. So our somatic cells, our body cells, are going to be primarily diploid. Reproductive cells are going to be haploid. Humans have 23 pairs of chromosomes. So it's 46 chromosomes in total, 23 pairs. Okay. And next we can talk about this overview of meiosis. And so I'm not going to go into too much detail because you've had this in a previous class. So if you have a good memory of this, that's great. If you need more review, please take that time. But I'll remind you that to get to daughter cells, we're going to get 4 daughter cells from 1 parent cell here. Okay, so when we go through meiosis, we go through essentially 2 cell divisions. So this cell divides into 2 cells, and each of those 2 cells divide into 2 more cells to produce a total of 4 cells from these 2 cells. And so if you remember your stages of cell division, you've got prophase, metaphase, anaphase, telophase happening twice to produce 4 daughter cells from that single parent cell in meiosis. So again, in humans, this is only happening in our gametes to produce our gamete cells that are eggs and sperm. Okay, so we start with the single diploid cell, 46 chromosome, each with duplicate sister chromatids. Then, those for the first meiotic division, you end up with 2 cells that are each 1N. So these cells are now haploid, but they still have their duplicate sister chromatid attached. In the second round of meiosis, you end up with 4 cells, each of which are haploid, each of which have 23 chromosomes with no sister chromatids. Okay. So really simplified, we can think about our single diploid cell with 46 chromosomes, dividing once, dividing twice, to end up with each gamete being unique, each gamete being haploid. Why are they unique? Because during this first set of meiosis, this first stage of meiosis here, we have crossing over, meaning that there are exchange of genetic material between sister chromatids, producing unique combinations in the gametes. Here's a look at that crossing over. So here's, for example, let's think about these as being your chromosome number 1. So here's a chromosome number 1, and each one is duplicated, right? You have your pair here. When these line up during that first meiosis, these homologous chromosomes can exchange bits of DNA. Okay, and now you have 4 different chromatid combinations, each of which would go into a different gamete, therefore ending up with 4 genetically distinct gametes from your single parent cell. So this crossing over produces a lot more variation than in our gametes. Okay, so lots of great meiosis videos you can watch to remind yourself of this process. The summary things you need to know are going to be that there's 2 rounds of cell division. Replication happens during prophase 1. Those tetrads form during metaphase 1 where you have crossing over occur, and that's really important for genetic variability. The result of meiosis being that there's 4 cells with 23 chromosomes each, and because of crossing over and this process of dividing a diploid cell into haploid cells, each gamete then is genetically different from the others. Okay, so now let's go into reproductive anatomy. We'll start with this system here, where if you have a gonad that is a testis, that testis is going to be inside the scrotum, and that's going to be associated with a bunch of tubules that make sperm and that make semen that allow exit through the penis. Okay, so remember this some is anatomy stuff. You have a single exit point here for semen and for urine, because here's the urinary bladder, and the urinary bladder and urethra is then going to also empty out here too. And you have an entry point from the testis, right, and through the ducts here, as sperm travel through these ducts and accumulate semen components that will then also empty into this common urethra here, okay? But remember that there is a little valve here and a valve here that prevents urine and semen from coming out at the same time. You're only going to have one or the other, okay? And then on the back end here, anus coming from the digestive tract. Okay, so review your anatomy, review all these different vesicles and ducts that exist and the glands that exist that help to produce semen. Okay, looking closer, looking into the testis, we can see these seminiferous tubules where the sperm are going to form. Okay, and then you can see here this epididymis where the sperm mature before they are ejaculated. Okay. And looking even closer, if we look inside the lumen of the seminiferous tubule, we can see the spermatozoa here being produced through that process of meiosis and eventually move into the epididymis for maturation. So the production of spermatozoa at puberty happens in the seminiferous tubules. It begins as spermatogonium that is enveloped in a Sertoli cell. So boys are actually born with many spermatogonia. The cell goes through mitosis then. It's making 2 identical cells, cell A and cell B. Cell A stays and cell B leaves and becomes a primary spermatocyte. The primary spermatocyte moves through tight junctions of Sertoli cells, then goes through meiosis 1 to become 2 secondary spermatocytes. So note there's that initial mitosis before you go into that meiosis. Okay, then it's going to go through meiosis 1 and meiosis 2 to become 4 early spermatids. And in the process of spermiogenesis, you get conversion of those spermatids to spermatozoa. At this point, they're going to lose the majority of their cytoplasm. And we can see these different stages of spermatogenesis then moving from this wall of the seminiferous tubule into the lumen. Okay, so here's a spermatogonium with 46 chromosomes. We said that divides into the primary spermatocytes, and then that will go through meiosis to become a secondary spermatocyte, a spermatid, and finally spermatozoa. Okay, so a couple of videos here where you can watch this process, but again, knowing that there's a mitosis at the beginning of this process that takes you from spermatogonia to primary spermatocyte, and then you go through those 2 meiotic divisions before you get your 4 spermatids. And spermiogenesis is that process of turning those 4 spermatids into spermatozoa, because now they've added this flagella so they can move and they can swim. Okay. A different way to look at this process, but the same thing happening here. Looking at this process happening inside that seminiferous tubule. Okay. And so what you find when you actually look at a seminiferous tubule then is you're going to find cells in these different stages of spermatogenesis. So you'll be able to see probably some spermatogonium, some of the mitotic divisions, the primary spermatocytes, and eventually these sperm here that are moving towards this lumen of the seminiferous tubule. And a mature sperm or an almost mature sperm is going to have this acrosome on the head, as well as a long flagella there too. Okay. So a mature spermatozoan is going to have a head inside this acrosome, a mid piece here with this little twirly bit around the middle, and then a principal piece and end piece here of this flagella tail. And when we're looking at the sperm actually fertilizing an egg, all these pieces become really important. Okay, this principal piece of the flagella is going to help it travel along the vaginal tract, up the fallopian tube to fertilize that egg. And this acrosome is going to have a really important role in actual fertilization of this egg. Okay, so what's happening here at fertilization? Well, in the vagina, only about 10% that make it into there survive. This is because the vagina is very acidic, and there's cervical mucus and uterine contractions also that are relatively inhospitable to sperm. And so there's a really kind of complex chemical interaction between sperm and vaginal mucus and vaginal acid. That's pretty interesting, and we're learning more and more about it as we think about things like IVF technology, right? So only about 10% of the sperm are going to survive. If those sperm enter a fallopian tube, then you can have them swimming towards a recently ovulated ovum. Okay, and so this process of sperm capacitation is when the sperm swims quickly toward the ovum, and that acrosome, that tip portion of the head, becomes fragile. Okay. When the sperm goes to penetrate the ova, it has to break through something called the corona radiata, and this is made of granulosa cells. We'll talk about that in the next lecture a bit. So the acrosome's primary job here is to release enzymes to allow penetration of the ovum. And then the sperm has to break through a zona pellucida, which is a layer that's made of glycoproteins. And finally, there's fusion of the plasma membrane of the sperm with the plasma membrane of the ovum. I want to note that you're only really going to be able to have a single sperm fully penetrate the egg. And so there's going to be chemistry at the level of these membranes that prevent polyspermia. They prevent more than one sperm from fertilizing that egg, because if you had more than one sperm, you could have trisomies. You could have 3 sets of chromosomes, and that's not going to be compatible with life. Now, certain trisomies are things like Down syndrome, trisomy 21, where you have 3 copies of chromosome 21. That can happen. But that's going to be resulting from a lack of separation of chromosomes during meiosis in the formation of sperm or eggs, not of multiple sperm fertilizing an egg. Because if you get multiple sperm fertilizing an egg, you have 3 full sets of chromosomes, and that's not going to be compatible with life and with development. Okay, so looking a little more closely at this acrosome reaction, here it is before activation. So here's the nucleus of the sperm inside the sperm head with the acrosome on top. Okay. When it binds to the zona pellucida, it stimulates fusion and exocytosis. So after the acrosomal reaction, you end up with then essentially a degraded acrosome with the plasma membrane a little bit more available here too, and the inner acrosomal membrane more available. Okay. So the nucleus of the sperm cell moves into the cytoplasm of the ovum, and very quickly, within milliseconds, changes occur in the ovum to black polyspermy. At that point, then, the ovum is going to quickly finish meiosis too. So it's actually hanging out here in arrested meiosis, and it's going to quickly finish meiosis too, and then the ovum and sperm chromosomes are going to combine, resulting in a diploid zygote. So diploid zygote, it's going to be 2N, 46 chromosomes, 2 sets of 23 chromosomes. I mentioned trisomy 21. There's also trisomies, quadrisomies of sex chromosomes that are also normal and happen and compatible with life, okay? But again, if you have 3N or 4N, where you have multiple sets of chromosomes, that's not going to be compatible with life. Okay, so another view of this fertilization process, the acrosome containing enzymes coming here, allowing degradation of the corona radiata and the zona pellucida until a sperm can reach this cell membrane surface here of the ovum, and eventually a single sperm cell nucleus moving inside the ovum. Okay. So I mentioned that sexual differentiation occurs because you have 2 of the 46 chromosomes determining sex. Okay. And this is traditionally taught as XY being male and X. Okay, so most textbooks is going to be said as XY equals male and XX equals female. This is often called genetic sex, though there's variation here. Okay, I'll talk about that a little bit. So when this happens, and in most cases at week 7, there's a sex-determining region of the Y chromosome, an SRY, that signals gonads to become testes, and then the testes are going to make testosterone and sperm. So if somebody is XX, they're going to lack this gene, the SRY gene, and so at week 9, the gonads are going to develop into ovaries. So I say typically here, and I say generally because there are a lot of variations here that can happen, right? Variations that are going to be variations in gonads, variations in external genitalia, internal genitalia, and therefore variations in sex. So this sex binary here is not real. There's more variation in human sex than is shown simply in your textbook. Okay. Here's a really interesting study I recently saw about this. A fertile woman with normal ovaries presents with a predominantly XY karyotype. So she looks like a woman, she is a woman, she has ovaries. In fact, she even has given birth. So she's fertile, okay? And she's given birth to an XY daughter with complete gonadal dysgenesis. All right, so when they actually went to go ahead and genotype the mother, she looks phenotypically typical woman, female. They found that she had a 46, that means 46 chromosomes, XY karyotype in the blood, 80% XY, and 20% just a single X chromosome in skin cells, and 93% XY, 6% X, and less than 1% XX in the ovary. So this is wild, right? Almost all of her body tissues are XY, XX in the ovary, fertile. And very commonly you'll see that these chromosomal variations in sex chromosomes are going to be described with infertility, but that's not always the case. And this is a really interesting example of what they call mosaicism, where your cells actually have different karyotypes, different chromosome compositions, depending on the kind of cell. And in this case, most of her cells are going to be XY, but she's developed as a woman, as a fertile woman, right? Okay, so I linked the article here because this is very, very interesting. Okay. So when XX, typically people are going to lack the SRY gene, although again, there's examples where you have transfer of the SRY gene onto an X chromosome, and then you might have male typical characteristics or testes developing in an XX person. Okay. But when somebody's XX and they lack the SRY gene, the gonads are going to develop into ovaries. When somebody's XY and their Y chromosome provides the SRY gene, the gonads are going to develop into testes. Okay. And then you can get follicular development in the ovaries or sperm development in the testes. Okay. So now we got to go back to a little bit of our endocrinology, and I want you to know and understand this figure. The hypothalamus is going to produce a hormone called GnRH. GnRH is going to tell the anterior pituitary to make FSH and LH. Those are going to tell the seminiferous tubules and the interstitial, the Leydig cells in the testes, to make two hormones. One is called inhibin, and one is called testosterone. And these hormones together are going to control sperm production and sperm maturation. Okay. So testosterone is produced by interstitial cells of the testes. It's an anabolic steroid that stimulates growth. And so you get an increase in muscle mass, a deeper voice, facial hair, and sweat glands when you have higher levels of testosterone. And this is going to be true in both males and females, people that are both XX and XY, is that testosterone is going to increase these things. Okay. It also signals sperm production in seminiferous tubules. And so now I want you to think about this case study. A 38-year-old male is upset about his low sperm count and visits a practitioner, quote unquote, who commonly advertises his miracle cures of sterility. All right, so the practitioner says, let's go ahead and inject you with testosterone. Let's treat your condition with megadoses of testosterone. The patients have a surge in libido, although sperm count goes even lower. Explain why. So what do you think should really be done in this case? What would a legitimate practitioner do? Come talk to me in office hours, and we can talk more about this issue here. And next time we'll get into part 2, reproductive physiology.